Hello everyone and welcome again to Biochem Bits. In this video, I'll be talking about the role of trace elements in metabolic catalysis. Metals are able to drive metabolism by acting as catalytic partners with enzymes, and they're able to set the stage for biochemical reactions to proceed. To understand this importance of metals like this trace elements, it's also good that we know what the atomic architecture is, and because this is what helps us to be able to perform that function of facilitating metabolic process or biochemical reactions. If you're familiar with the periodic table, you know that transition metals are also known as trace elements, and this the majority of them are known as trace elements. And these elements are the ones that occupy the atomic number 21 to 30, the ones that are very common, and they have this unique d-orbitals. They occupy the d-orbitals, and this enable them to be able to have this flexibility in coordination of electrons, being able to pass electron around, offering them that geometric capacity hanging on to in the active site of the enzymes that they are partnering with structurally. So you see the 4s2, 4s1, 4s1, and those outer electron flexibility is one of the reasons why it's able to perform this cofactor activity. And so how does this relate to catalysis? How does it partner with enzyme to facilitate metabolic process? So when we think of metabolism, we often look at it as enzyme doing all the work. So this is telling us that the role of cofactor in facilitating metabolic or any biochemical reaction is also very, very crucial. As much as we know that enzyme facilitates or increase the rate of reaction or by lowering the activation energy, the role of these cofactors, these metals, is very, very important, and we'll get to see the biochemical reactions that happen and how they are able to act as structural partners to make this reaction go ahead. Because most metabolic molecules carry very strong negative charge. If we try to bring them together in a tiny enzyme pocket, they may repel each other. Like charges repel, unlike attract. So because of this, ion help us to solve this problem by beautifully acting as a Lewis acid and neutralizing those negative charges and stabilizing the entire reaction environment that will allow the reaction to progress. I liken this to when you have a machine that has maybe some drill bits. So the trace element acting as a cofactor is serving as the drill bit that provides that reactive chemical edge. If looking at that picture that you can see on the screen. Now, because of these diverse chemical properties that they have as transition metal, they are able to form part of the biological toolkit for reaction to progress. Nature generally divides them or their cofactor activity into two. Their ability, one is the Lewis acid charge stabilization mechanism, and the other is the redox shuffling mechanism. So looking at the Lewis acid charge stabilization mechanism, the metal ion like copper, zinc, cobalt, selenium, iron, they are very well positively charged, and they're able to neutralize the charges of the substrates, which are, as I mentioned earlier, are highly negatively charged. So this stabilization allows substrates to be able to fit properly inside that enzyme, and because there is no repulsion, there is attraction, and then is able to pull those substrates together, forming that lock and key that is required for the lowering of the activation energy for the reaction to progress. And so this reduction in the electrostatic repulsion allows those reactants to be in close proximity. And you know that one of the factors or one of the things that makes reactions progress is proximity, their ability to come together. You remember factors affecting rate of reaction, coalition, the distance between interacting molecules is also that. Those have some elementary chemistry there. Now, this precise positioning is what facilitates. It's not that it is the cofactor that is performing the function, but it is the ability to bring this thing together in that Lewis acid stabilization, acting as a proton donor by releasing proton to the negatively charged substrate, making them to come together, fusing them together, and then the reaction is able to progress. On another hand, we have the redox shuttle. How does it do this? Metals like zinc, like copper, like iron are able to act as redox intermediate. They act as redox shuttle. They utilize their shifting outer electron. You can see that the s orbital that has one, that has two in the previous slide. Their outer electron shell is able to cycle reversibly between oxidative state and reduced state. For example, you remember iron can be in Fe2 plus state or Fe3 plus state. That is reduced or states and oxidized states. So driving their transition between those two states helps to drive continuous transfer of electron throughout the metabolic pathway, and this will facilitate the metabolic catalysis, allowing it to lower the activation energy, and then it can progress. Okay, so when we look at this very other example in the case of carbonic anhydrase, seeing how the presence of zinc helps carbonic anhydrase to convert toxic carbon dioxide into gas soluble and safe bicarbonate. This process is very, very important, and it happens very fast. Deep inside the active site, the zinc is held on by a triad of histidine amino acids, and in some cases it could be held by aspartate 162, histidine 220, and cysteine 160. But the more common one that I'm focusing on is the one the one that has histidine as the active side amino acid to which it is bound. Histidine 119, histidine 96, and histidine 94. So what zinc does is to bind a nearby water molecule by bringing it down from the 9.6 to 7.0 pH, and then this will simply trip a proton away. 9.6 pKa to 7.0 pKa, and then this will make it to generate to make the water molecule to split, leaving a highly nucleophilic species. If you remember the substitution, the movement between electrophilic substitution and nucleophilic substitution in elementary organic chemistry, you know that there is a movement or shift between those bonds that allows proton then to be released, and then you have a highly nucleophilic subspecies being released. And in this case, it is hydroxide. The presence of the hydroxide facilitates the attracts the carbon dioxide, and then it quickly binds to the carbon dioxide to form the bicarbonate. The hydrogen carbonate, the HCO3, as you'll be seeing in the next slide. CO2 combines with water to form the HCO3. Now, moving from charge stabilization now, that zinc function in carbonic anhydrase reaction is a plain example of charge stabilization Lewis acid mechanism through which cofactors act to facilitate metabolic reaction. Now, let's go to the electron transfer example. And this we'll see in the case of iron and copper, how they are able to drive mitochondrial power grid in the electron transport chain. We all know that during cellular respiration, some complexes are involved, and they are present in the mitochondria, acting as the electron acceptor or electron transfer within the electron transport chain. So here, iron acts as the ultimate electron shuffle. It shuffles between those complex, and it irreversibly cycles between ion 2 and ion 3. As you can see on the screen, it's becoming reduced and oxidized, passing the electron to the next one. And it also works in tandem with copper. Copper is moving between copper 2 and copper 1, and then finally pass the electron to the final electron acceptor of the electron transport chain, which is oxygen. Now, another scenario that I want you to pay attention to is the activity of galactose oxidase, which is the enzyme that converts primary alcohol to their aldehyde, their corresponding aldehyde, and then release H2O2, that's peroxide, which is also acted upon by some an oxidative enzyme. Normally, for this reaction to progress, there is a need for an executing complex a combined 2 oxidative reaction in such a way that you have it as a multi-step metal cluster. So we have it, first one will happen, second one will have to happen. Instead of this happening, the enzyme couples a single copper ion with a modified amino acid within its active site. The copper ion and the radical are able to each handle a single electron transfer simultaneously, allowing the enzyme to be lightweight, and then is able to seamlessly execute the complex organic chemistry that is happening there. You can see from what you're seeing on the screen, you can see that this is very complex kind of. So the activated or the radical thyroxine there is the thyroxine 272. Thyroxine 272 is now acting in tandem with copper to synchronize the 2 electron transfer that is required for that aldehyde to be produced from the primary alcohol in response to the catalysis of galactose oxidase. Now let's look at those very vital catalytic trace elements that we know. And just to refresh our memory, we know of selenium, which is very important in oxidative processes, and though it's required in minute quantity, but we know that it facilitates a lot of reaction, allows flow, mop-up of free radicals, and participates in other very important metabolic processes. One of it is the one that is identified in glutathione peroxidase that reduces hydrogen peroxide to water and oxygen. Another one is the one I mentioned earlier, saying within carbonic anhydrase, and you know what the physiological significance of this is. It will help us to balance the blood pH, as well as increase or help facilitate respiration. Iron which uses its redox cycling mechanism. It aids aerobic respiration and also help in the production of energy unit, energy currency, which is ATP. Copper help facilitate the final step of electron transport chain, which is very, 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 very important. That's why deficiency of some of these trace elements could be lethal, could be life-threatening, because they participate in very, very important biochemical processes in the body. Manganese is also a very important trace element that is found as a primary driver of our photosynthesis. It's majorly found in the photosystem in plants, and that's why it helps in the splitting of water molecule. You remember the dark phase and the light phase of photosynthesis. The first thing that has to happen is the photolysis, the splitting of the water molecule, so that the hydroxyl ion will be available for the next step. Yeah, so finally, this is just like a wrap up. Every of these trace elements is very important for metabolism to happen, and metals allows this reactivity to happen very precisely, stabilizing the substrate and accelerating reaction. And so the flow of energy through the cell becomes right, and then they are also able to help with DNA replication, ATP synthesis, neurotransmission, detoxification, and I think they form quite a major architect of metabolic speed and its efficiency. So in summary, metal, this trace elements, these metals acting as cofactor is not just about helping with catalysis alone. It's also about being able to activate, in some cases, accelerate the chemistry, biochemical reaction, and help to also make some of this reaction to work in synchrony. So thank you so much, everyone, for listening to this video. I hope it's not too long. And if you have any comments, please put it in the chat. If you have any suggestion for the next topic you want me to talk about, please put it in the chat as well. And if you have more insight on this topic, you can also put it in the chat. People will come and learn. We're just helping ourselves to learn and bring more people more to biochemistry, to understand the chemistry of life and be able to apply some of these mechanisms to our studies as well as to our daily life. Thank you.